Hello, and thank you for coming to my presentation on the mechanochemical comparison between hydrogel and silicone gels used for studying stiffness sensing by adherent cells. Many hours of work went into this project, and it was contributed to by many co-authors, including myself, Kathleen Heusser, Nikhil Mittal, Mohannes Chatterkar, Zongtian Zhang, Karar Alafari, Dr. Jeffrey Allen, Dr. Bruce Lee, Dr. Fei Long, and Dr. Sang-Yin Han. We issue from the Department of Biomedical Engineering and the Department of Mechanical Engineering at Michigan Tech. For a bit of background, the extracellular matrix, or ECM, stiffness affects cellular adhesion and spreading by cell force sensing mechanisms. Investigation of these mechanisms has yielded some contradictory results, and we think that the different types of gel used in the studies may be the culprit. On the left, you see a figure from a study by Engler et al. In 2004. Cell area increases with increasing stiffness of the gel. This was done using polyacrylamide gel. On the right, you see a study by the Han lab using Qgel recently. And this exhibits a biphasic trend. So after a certain threshold stiffness, the cell spread decreases instead of increasing as gel gets stiffer. Our methods including making polyacrylamide gel of different stiffnesses by using different ratios of ingredients, and the same thing with Qgel. Quantum Silicone's Qgel 920 was made with different ratios of components A and B, and these affect the stiffness. We used a DHR2 hybrid rheometer below, and that applied a known shear rotational strain to the gel and measured its deformation. This quantified its stiffness. We also used a sessile drop technique to measure the contact angle of water on each gel, and this showed us the different hydrophobicities or hydrophilicities of each gel. To measure the attractive capability or stickiness and stiffness of the Qgel, we used atomic force microscopy or AFM, but found it necessary to use BSA treatment, bovine serum albumin, first. Our first finding with contact angle measurement was that Qgel is far more hydrophobic than polyacrylamide gel. Consistently, Qgel had a contact angle of 120 to 130 degrees across multiple stiffnesses, as you can see in the left figure below. Polyacrylamide gel showed a far more hydrophilic characteristic with a contact angle of 20 to 30 degrees, and this changed based on stiffness. We used a Mann-Whitney U-test to establish that more stiff polyacrylamide gel definitely leads to a more hydrophilic gel or a lower contact angle. Also, we plasma treated Qgels for varying times from 30 seconds to 2 minutes and found that 30 seconds was most successful in lowering the contact angle or making Qgels less hydrophobic. This showed that Qgel hydrophobicity is adjustable. Our next finding was that atomic force microscopy only works when the surface charge of the Qgel is changed. So originally, AFM showed a huge attractive force to the cantilever used from the Qgel, bending it, in fact, when the cantilever was nowhere near the surface of the gel. Then, after coating it with bovine serum albumin for 1 hour, this changed the hydrophobicity of the Qgel, making it more hydrophilic. It remained flat, and the cantilever was able to measure the attractive force as normal rather than being attracted to it from the first moment of measurement. Our rheometry results gave us the storage modulus G prime for Qgel, and this essentially was quantifying the stiffness of the gel ratios that we were able to make. A paper by Gutierrez et al. From 2011 has already calculated stiffness of Qgel using a different technique. Our values were a little bit different from theirs, but seemed to come to a pretty uniform plateau as seen in the bottom figure at about a 10-hour time, because this was a time sweep where the variable was the number of minutes or seconds that the gel had been being moved around rather than trying to change the stress or change the strain. In the future, we will use plasma-treated and untreated Qgels to test hydrophobicity's effect on cell spreading. We anticipate seeing that hydrophobicity dominates spreading. We'd like to thank Dr. Faith Morrison and Dr. Paul Bergstrom for equipment assistance, and Portage Health Foundation and the National Institutes of Health for support. Thank you.